Let's refine it further. Let's refine it further. We'll make it mad. We lead the way in the reptile world. We're here to stay. Double clutch, where passion meets. From the start to the top, we can't be beat. Double clutch, we're making that double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Double clutch with Mike and Matt. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Let's refine it further. We Mike and Matt, lead the way. Let's refine it further. We're here to stay. Double clutch, we lead the pack. Double clutch, always on track. Double clutch, we lead the pack. Double clutch, always on track. Double clutch, we're making that. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Let's refine it further. Let's refine it further. Always on track. We'll make it back. With the clutch, double clutch, we lead the pack. Double clutch, always on track. Double clutch, we lead the pack. Double clutch, always on track.
Yo, what's good, bro?
What up, baby? It's double clutch time, man. You already know what it is, baby. Time to tear it up, baby. Let's go.
Yes,
Let's go, man.
Yes, sir, man. Been looking forward to this, man. We're gonna tap into a real subject, man. It's gonna be tight.
Yes. This is gonna be good. This is an interesting one for sure for me. Because you know what happened with me last year with that white leg. So I got scared about a bunch of things.
Yeah, man.
So
it's so much to tap into with this, man. It's probably a lot of misinformation out there. I know myself, I got a ton to learn. So, man, I'm definitely looking forward to it and looking forward to picking the brain of our guest tonight, man. How your week been going so far?
Oh, man, it's been going good. If I keep rubbing my eyes, man, it's because I got drywall and all kinds of dust and fiberglass in my eyes from being a construction worker, like I was telling you all day today. Basically trying to get ready to blow this wall out. So I'm building a wall on the other side of it. Took down the drywall in the garage so I can insulate it and then put some more drywall up. Then I'm gonna just blow this wall out and have my electrician guy come and move all the electrical where it needs to be. But yeah, so I'm sinuses is going crazy, dust is everywhere. Yeah, so it's been a crazy day, man. What about you?
Yeah, man, it's been a crazy day for me too, man. Been ripping and running, getting prepared for Daytona, man. Just running through my setup, making sure I'm like crossing my T's, dotting my I's, all that good stuff. Had to make some last minute runs to get a few supplies and stuff like that, man. Just really trying to make sure we super prepared for this Daytona. So inevitably, it'll probably be something that slipped through the cracks. So it'll be a learning experience overall because it's our first time doing something like this. But, man, really looking forward to it, looking forward to seeing everybody, man, and really getting on the road, taking the show on the road, man, just trying to grow. You know what I mean?
Yeah, I'm excited about that, bro. Daytona's gonna be crazy. That's for sure. It's gonna be crazy. I think the only show, other than the ones that we've been to, like here on the West Coast, SAC, Reading, all those kind of stuff over here. We went to Tinley and then now Daytona this year, so hopefully we'll be able to go out to Texas. But we're excited about Daytona, man. We looking forward to it for sure.
Yeah, it's gonna be a blast, man. It's gonna be a blast. It's crazy, man. Like I feel like I've been knowing you forever, but technically I haven't met you in person, but feel like I've been knowing you forever.
Right, man. Shoot, I ain't met me, Justin, you, Jaffe. Shoot, nobody. So it's crazy. It's crazy though what you could do with communication now with doggone phones and FaceTime and all that kind of stuff. So it's wild.
Yeah, 100%, man. But yeah, definitely, man. Looking forward to Daytona, man. It's gonna be up in just a couple weeks, man. Not too long from now.
We're excited.
Yeah, man. But so without further ado, definitely want to get the guest out here, man. So, because this is what the people came from to learn, man. So, hey, we're
Yeah,
gonna bring out Dr. Stephen Tillis. We're gonna let him tell you a little bit about himself. Then we're
the
gonna hop into
doctor.
the conversation. The doctor, the good doctor.
Yeah.
Yeah, UF alum, University of Florida. Go
Oh
get
God.
Awesome. Thank you guys for having me.
Yes, sir, man.
Oh
Well,
yeah, man.
man, thank you for coming.
Yeah, no problem. Absolutely.
Cool. So, man, give everybody an introduction. Tell them about yourself, man, so they know about your background.
Yeah. I've been breeding snakes since 2009. Did my undergrad at University of Florida in wildlife ecology and conservation. Was a full-time reptile breeder for a couple years. Have done some consulting for some very large python colonies here in Florida. Ended up losing a good portion of my blood python colony to NIDOVIRUS, or what the scientific community would call serpentovirus, and basically put me out of business. At the same time, I was able to get my foot in the door at a lab here at the University of Florida's College of Veterinary Medicine to study NIDOVIRUS. And so, yeah, I was kind of able to flip that into the research that I was able to do. And then in the middle of my PhD, the state changed all the laws about most of the large constrictors that I kept. And so I had to build an educational facility. And so I also have like half the world's python species here, with 4 different buildings, with each building kind of a different theme. So I have one which is like a biosecurity themed building with about half the world's python species. I have a display building with a lot of my really crazy ideas for exhibiting animals. I have a rodent building. I have a commercial production building, which is where I breed blood pythons and short-tailed pythons. And I still do work in the lab. So yeah, I have kind of a lot going on, but all of it is kind of generally reptile themed, just through a bunch of different lenses.
Nice. Yeah, man, that's quite an impressive background for sure.
Yeah, that's
It's been
wild.
a long road.
So were you always into reptiles, or did you just kind of like at what point did you decide I want to work with reptiles, and what kind of drove that?
Oh, it definitely started very young. And so, I remember even as like a 5-year-old finding like wild rat snake in the yard and like, keeping it in a pretzel jar, for a few days. But it wasn't until probably like 2008, 2009 when Barchek really started pushing a lot of his videos online that the idea of like, I could do that popped into my head. And so, yeah, that was basically right in the middle of high school. I sold my Legos on Craigslist and bought a pair of ball pythons and started breeding from there.
Nice, man. I think I cut you off. You was about to ask a question.
No. That's good. You got him right on the right track, because he said something about 2009 breeding. I was just gonna see what he was breeding back then, or what he was doing, because he don't look like. I mean, how old are you?
I just turned 30 this year. Yeah, I've been doing it. Like I said, I've been doing it since I was very little. It's funny.
Yeah.
I walk around expos, and a lot of the people are like, the vendors will remember me from when I was a kid. Walking around. But yeah, no. So this should take you back in the time capsule. So the first ball python clutch I ever produced was a male pinstripe ball python to a female normal, and I produced 1 female pinstripe from that clutch, and that female was worth about $800 at the time, and so I was able to trade that for like 10 adult normal females, and then the next year produced like I don't know 15 pinstripes when they were still worth about like $400 to $500 each. So it was back when like the incomplete dominant math worked out like that, where like you really could double your money in a weird way like that for a short period of time for something as simple as breeding a single gene male incomplete dominant to a single normal female and build a whole collection off of that. But yeah, so that was yeah.
Yeah, every
Yeah,
time I
that's
breed
wild.
pinstripe, I hit like 7 out of 8 pinstripes.
No, it was actually a joke there for a while because I just had a hard time stop producing pinstripes. So it's still one of my favorite genes, but like it just I don't know. There was it was so much harder to get stuff back then. Like it really
Yeah.
was. Like So yeah.
And you were in high school when all this was going on?
Yep. Yeah. So I've been
Okay.
doing that was sophomore year of high school was when I produced my
Yeah.
first clutch, and then from there it grew to mostly focused on pythons. But I've worked with everything, and even at the zoo I have here, I have like Aldabran giant tortoises and big parrots and branch-tailed skinks, all sorts of stuff.
Okay, yeah, we definitely gonna get into all that kind of stuff because I like different variety of stuff. I got a whole bunch of stuff. So yeah, you talked
I
about
really specialize in importing and establishing that kind of stuff. So like the species that I keep, half the world's python species, those half of the world's python species are the ones kind of not commonly bred in captivity. So basically all the species not in my colony are ones that are either almost unobtainable or so readily obtainable that it's like kind of not really worth me trying to focus in on them.
Right. Okay.
Gotcha. So speak to some of those species that you're keeping and kind of speak to the process of like acquiring those animals, especially like the ones that are like super hard to obtain.
Yeah, so that part of it has been kind of like a probably 10-year journey at this point of working with wild caught stuff and just learning how to read it and get it in and when to take the appropriate steps of treating it and when not to and when to just step back. And so like one of the species I'm most proud of working with here are the Halmahera pythons. And so that is a species of python that has been bred in captivity fewer times than any other python species. And I was able to hit on a clutch this year, and I'm waiting for them to hatch out like within the next day or two. I'm very excited. But this species is notorious to keep in captivity because they're just so shy that they just stress out and die. And so like the group of animals that I had them in 2 years inside of entirely enclosed boxes. So like no glass front, like the front on it was solid except for a little peephole that I could look in there. And so I didn't touch those animals for like 2 years. All the stuff that's going into that biosecure building went through a year-long quarantine with multiple NIDOVIRUS tests throughout. That biosecure building has like an air scrubber and like a entry procedure and sanitation routine. It's just any idea I had, I kind of took it to 10 on that. And so like a lot of the species go through a very long quarantine with a lot of testing. Then they get put in that building, and that's kind of like my genetic vault of obscure pythons. And so the Halmahera pythons, after a very long quarantine and basically never letting them see me look at them because that's just how shy they were, they finally kind of settled in where I could start keeping them like a normal python. And from there, once they were able to live in a normal snake cage without really feeling all that stressed out, I started putting them together in a group, and I ended up getting a $80 marijuana grow tent off Amazon and set them up in a big communal setup and got a clutch the first year. So it's a lot of it is just working with these species or just pythons in general for just so long that it's just.
So these guys
work with them.
They stay in a community group, kind of like garter snakes or whatever, like that kind of deal.
Not necessarily. There's not really a recipe for breeding them one way or the other. And so, a lot of what I do at this facility is kind of challenging both the norms of the standards of modern herpetoculture, as well as, so in some buildings, I have a traditional setup of a commercial breeding facility, which is one snake per tub, like your classic snake setup. But, I've been doing this so long that I've really kind of given myself permission to experiment with what is acceptable or not. And so I have ball pythons in communal setups that have been living in communal setups for 5 years at this point. And so for a lot of species, the rules aren't necessarily as firmly written as herpetoculture necessarily considers them to be. And a lot of times keeping these species requires a little bit of experimentation, and that experimentation also kind of requires just the fact that I've been doing this for 15 plus years and kind of know where the limits are, you know, because you will hit the limits on the animals if you push them in these scenarios. And so but at the same point, a lot of these species, there's really no other way to work with them. And if it works, it works, and it works for me. And so, like, yeah, I don't know.
Nice. That was smart, like how you did the one species. I don't want to try to pronounce it because I probably pronounce it wrong, but basically, essentially, like tricking them into not feeling the need to be shy because they really can see that you were observing them and that you were like right there, and until they got comfortable. And then once they got comfortable, then you could put them in like a normal enclosure, and then you were successful with breeding. Is that something that you were like taught by a mentor, or is that just something you figured out? What's up with that?
Is that those?
Yes. Yeah. That's those. Yeah.
Okay.
Yeah. And so they have been bred in captivity. My clutch is the 9th one that has ever been produced in captivity.
Oh, nice. Yeah, that is nice.
Yeah, that's pretty wild.
Yeah, they're a very unique python. All pythons are unique, so you know. But so that species is named after Tracy Barker, and so she does a lot of VPI axanthic. That's Tracy. So that is Somalia tracyi, or basically Tracy's python. You know.
Okay, yeah, that's nice. That's real nice.
Yeah.
It's kind of got a little bit of carpet python vibe, just a tad bit.
Right, that's what it
they're from
reminded
that
me of.
general part of the world, but longer than a carpet python, and just like I said, about orders of magnitude more fragile.
Oh, so they get big.
Well, big but slender. Like imagine like a 12-foot Amazon tree boa. Like, you know, like it's just weirdly proportioned. Like it'll be like the size of a small to average adult ball python, and then it'll unfurl and just keep going. So there's a weird snake. Scrub pythons. So scrub pythons are unique in that they have a strike range that's about 75% the length of their body. And so the first like 6 months I was keeping them, I kept getting the shit bit out of me because I kept underestimating their strike range. And I've never seen a snake that'll just throw themselves at you the same way a scrub python will. And they're so long that if you are touching them, you are within the strike range. And so there's no like safe way to handle them. It's really just kind of like working with them.
Rob from Rob's Creeping It Real, he works a lot with the scrub pythons too, right?
Yeah. I've worked a lot with Rob. Yeah.
Nice. All right, boss. So tell us about NIDOVIRUS, right? For those, like, break down the basics, right? For those who either don't know or maybe they've heard misinformation, kind of give us the rundown on NIDOVIRUS and how it affects reptiles.
Yeah, so I mean the biggest thing, not even the biggest thing, but like, you know, even right off the bat, calling it NIDOVIRUS is sort of like an interesting sticking point I still hear in the hobby. And so I refer to them both as nidoviruses or serpentoviruses because a lot of people in the hobby still just default to them being nidoviruses. Mm-hmm. But like nidovirus is a very, very, very general term. So that's like as zoomed out as you can go. It's like calling a python a reptile.
Okay.
Like basically like coronavirus is a nidovirus. And so when these viruses were first discovered, we didn't know what they were. And so they called them a new reptile nidovirus because it was the most general thing they could call them before they had anything else to call them. Now, at this point, we've really done a good job of classifying these viruses and understanding them more. And we've given them a new designation, and that is serpentovirus. And so it's not incorrect to call them nidovirus. I still call them both. And everyone in the hobby still calls them nidoviruses. So it's, you know, I'm not going to correct the record on that. And make any progress, you know, doing that. But at this point, like in terms of having people understand what we're talking about, like this group of viruses that we frequently see causing respiratory infection in pythons and other reptiles are serpentoviruses, which is a group of 7 different genera of virus that affect reptiles from skinks to turtles to mostly snakes, mostly pythons, but with colubrids. We're actually just working on a study that is putting out a new genus of these viruses and a new clade of reptile, but I can't quite talk about that yet, but in like a month I could. But like, you know, basically it's a very, very, very big group of viruses in a very, very big group of viruses in a very, very big group of viruses, you know? So it just, when people talk about NIDOVIRUS, they think we're talking about one thing, and we're really talking about 7 genera with hundreds of species within that, hundreds of viral species within that. And so like, it's in some ways overwhelming, but in other ways, like, really is just about understanding the questions you're asking and kind of setting the expectation of like what the real risk is for a lot of these things. And so like, one of the studies that I was able to do was looking at the Burmese pythons down in South Florida, and we basically found stupid high viral prevalence without any sign of disease. And that seems to be kind of a hallmark of these respiratory diseases. And so like, in a natural population and in most captive snake populations that exist, like, the prevalence of these viruses is very high in captive populations, and we are starting to understand in wild populations as well. The breakdown between the presence of virus and the development of disease is kind of a complicated thing that we don't really understand. And so like, a lot of the conversation is like, these viruses can cause a lot of problems under some sets of circumstances, but these viruses are also very prevalent and don't cause problems most of the time if you're using kind of common sense and good biosecurity. And so it's kind of a nuanced conversation about risk and disease and diversity and how good a test is going to be and how wide a diversity of viruses is it going to pick up. And so like, it's almost like we're starting to, so like, even just in green tree pythons, there's probably like dozens and dozens of different viruses, and those viruses can be pretty different genetically. And so making a test to pick up all these viruses is very, very difficult. But if that test is picking up enough of the bad viruses and you avoid the circumstances which create stressed out snakes that cause disease, the risk of these diseases are also kind of mitigated. And so it's like, it's unfortunately, it kind of has a lot of the same echoes as like a lot of the COVID conversations went, and we all kind of know those didn't go great. And so
that's what it sounded like to me.
Yeah, but it's like, you know, it's like a lot of it is just common sense and understanding that like there are circumstances in which these are pathogens that can be very serious and have very serious complications. And there's also most of the rest of the time when a lot of times the risk is pretty balanced in healthy populations and kept under reasonable set of, you know, non-stressful circumstances. And so it's like, so basically there's a lot of factors that go into it, and like testing is very important, but also, you know, people can get like just really freaked out over a positive test without realizing that a positive test is like probably going to be the present at least some level in most populations, especially if you've ever seen even the most minimal respiratory infection in a python.
Okay, let's dive into that. Let's dive into that, because I
Real
feel
quick
like,
shout out though to the homie Big Ed from Static Morphs. Appreciate
oh yeah,
that homie 262 in the building, man. Appreciate the support, man. This dude he support all the time, man. So really appreciate that. Big shouts
Most
out to
definitely.
you.
Is
Go
that
ahead, my bad.
is that our first is that our very first, what's the name for this show?
No, we got one before from Arvin.
Oh, we did. Oh,
Yeah.
okay. All right. Well, okay. So I just want to dive into that a little bit because I've been seeing a lot of stuff because we brought you on, wanted to dive into this subject because I just been seeing. I mean, a few years ago, the whole Nido stuff was running rampant or whatever. Everybody run to the hills, Nido's here, whatever. And then I'm starting to see it circulate back again on Facebook. A lot of people are talking about if you're not getting your animals tested before you ship it out to somebody to be able to provide them with a negative test, then you're not a good breeder. Stuff like just outlandish stuff like that, right?
Yeah. Well, so what I've tried to do is kind of update my morph market terms in terms of like how I understand these viruses to be and what's a realistic expectation. And so like a lot of times in so one of the many challenges and one of the many limitations of testing is that a snake has to be shedding enough virus to detect on a test as positive. And if a snake is happy and warm and not stressed, and in my snake room, it might have virus in it that is at such a low level that I can test it as much as I want, and it's not shedding enough that it's ever going to test positive. But I take that same animal and I ship it to someone, and it's stressed from that shipping, and then it might have virus start to come to the surface there. And so the best time to test an animal is receiving, not before you ship it out. Because if I can take, if an animal is happy before it's shipped out, you almost need that stressor to bring low-level virus to the surface if you want your best chance at getting a positive test. And so like for me, my guarantee is that I can guarantee a negative test on arrival, which is to say that when you get that box and open the box and swab the test and send that out, I think within about, so it takes about 24 to 48 hours for one cycle of growth for these viruses. And so that means if I ship a snake and you have virus in your colony and you're doing no quarantine and just slap it in there, if you already have virus in your colony, you might have contaminated the snake I sent you within 48 hours. And so if you test it before that window, and it comes up positive, then I can say, okay, that snake probably was positive when it left here. It was just such a low level that I wouldn't be able to detect it. And so in that case, yeah, please send that animal back. I'll replace it. But it is also important to know that, like, and then now the hard part is what to do with that animal, because that animal, even though it's virus positive, if it's happy, it's not shedding enough virus to really even test positive under normal scenarios. And so like, it just, what do you do with those animals? And so like, I kind of have this virus neutral colony, which is where I keep my adult breeders. And in this colony, like, there probably is serpentovirus or nidovirus, but I can't find it unless I have a reason to find it, which is to say that like an animal is pregnant and gets like mildly stressed, and then it'll get like a mild resp. Then maybe I can sometimes get a positive test out of those animals, but I even haven't had that happen in a long time now. But like I kept that adult colony closed for about 5 years and ended up pulling a new subgenus of virus out of a blood python in that colony randomly years later. And so it's like, I don't even know what to tell people at that point. It's like, I did everything right, kept that colony closed for 5 years. And not only was there a virus in there, it was like a new genus of virus. And so like the diversity is just so extreme that like, you're never going to detect make a test that can pick all of them under all the set of circumstances. And so like, if your expectation is squeaky clean, sterile, zero virus, I'm going to do everything I can. Like, even if you do that, you probably won't end up with 100% virus free. Like, I've done everything I can to make a building that is as 100% virus free as I can. And I still expect something somewhere somehow will slip through a crack. But I'm also gambling that that's not going to cause me any problems. That's just part of the microbiome of these animals. And so it is literally not possible to culture an animal without also culturing its microbiome. And so these pathogens, we are starting to understand, just exist kind of harmlessly in the background of these wild populations, and typically don't cause problems unless they do. And so the conversation is just as much about what are the scenarios that cause problems than, virus, no virus, because it doesn't necessarily boil down to that simple. And like, if you want to boil it down to that simple, you're going to have to end up making some very hard decisions about your colony when you end up finding that there's more virus in it or more pathogen than you thought. And so for me, my solution, for my serpentivirus free colony, which I consider that a specific pathogen free, which is a designation of saying that I can guarantee the animals that I sell are specific pathogen free for serpentivirus, because that's the pathogen I know how to look for. But like, that's just the pathogen we've heard about the most recent, and the list will be miles long because the microbiome is miles long. And so some of these are very important under some set of circumstances, and a lot of it is just kind of very complicated otherwise. But it's also a very fine line to walk, because I don't want to sound like I'm minimizing these viruses. I killed $250,000 of blood pythons that were positive for these viruses, and so like it's a very serious scenario. But again, I also feel like a long quarantine, appropriate testing, appropriate expectations, appropriate reactions when you see animals develop illness, and you kind of end up okay. Like, there's a reason that 30% of the Burmese pythons we found in the wild are positive for these viruses, and 30% of the Burmese pythons in the wild aren't out there dying of respiratory infection. Just as there's a reason that all of the python studies we see in Europe and the US find a prevalence of like 20 to 30% in adult pythons, and we don't see 20 to 30% of adult pythons out there dying of respiratory infection. And so it's a nuanced conversation. So there's a question I saw about can it be passed through the air or only by contact? And a lot of that we really don't know, and there's not a great way to find out. Our best guess is oral fecal via contact, but it also is probably very much dependent on viral load. And so like a snake that is a very, very, very low level positive that might not even detect as positive on some tests is probably going to require a tremendous amount of contact to pass along that virus to another snake, if it ever does at all. And that's probably how the virus operates in a normal, healthy animal and through normal, healthy population. But by the time you end up with a very sick animal that is stressed for whatever reason, has some sort of secondary bacterial thing or a fungal thing that's working in synergy with this virus, or vice versa, we really don't know. But basically, the sicker the animal is, the more viral replication is probably going on. And the more viral replication, the less contact it would take, because it's cranking out. Sometimes, that's the thing with viruses, is that they work in exponential growth, and we just don't think of in exponential growth naturally. And so like a sick snake might be producing billions and billions and billions of times more virus than a healthy snake. And so like, airborne or not, like it's just maybe a fly is enough to carry it over in that set of circumstances. But we really don't know. Like, this is just me spitballing at potential scenarios. But like, for me, I get scared of serpentivirus when I see sick animals. Sick snakes make sick snakes. And so when I see an animal develop even the tiniest little bit of something, I will remove it from the colony as soon as possible. But in a healthy, animal with just a positive test, like green tree pythons are a great way to talk about this, because like, I have green tree pythons that I've had for like a decade that have never shown any sign of illness, and I end up testing and are positive. And it's like, this animal's clearly always been positive. It's never passed along its virus to any other animal in the colony. I've been bred it multiple times, and none of the other green tree pythons came up positive. So like, it's not passing along its virus, yet the virus is there. So what does that mean? Who knows? And that's just one virus in one green tree python. in a group of viruses that is really good at messing and, making errors replicating its genome. Yeah, so I absolutely think that we really don't know the amount of viral load it takes to pass along from one snake to the other. And so it's probably hard to say like what's the bare minimum it would take to pass it along. But it's more just like the more problems you see in the animal, the more likely it is to pass along those problems to something else.
So Billy's question here about an animal with a low viral load or no symptoms, you put a rodent in the tub with it, it doesn't eat the rodent. You put it in the tub with another snake. Can that, in theory, pass it along?
Yeah, absolutely. Yeah. I mean, again, it really depends on the scenario. Like, again, if there's a low viral load animal and there's just not a whole lot of contamination on the rat, then like that might not pass it along. But like, I have no reason to say it would or wouldn't, other than just to say it would be way, way, way more likely to happen in a sick animal, because we know viral replication is going to go up. And when viral replication goes up, viral mutation goes up. And when mutation goes up, you end up with these really scary viruses that do kill colonies. Because as soon as you change the equation from can this persist at a low level without harming the animal to can this spread as quickly as possible, our snake collections start ending up being like a big bonfire waiting to go up in flames. We have a lot of very dense populations of animals kept very closely together with sometimes different levels of biosecurity. And so like a lot of that just depends on risk management and how you're spreading it out in the colony. And so like I live in the real world with real economics. And so like if you're going to put in a rat with a snake and it doesn't eat it, and you want to say, is this rat safe to feed? I don't know the answer to that. But if it is that important to recover that cost, then feed it to an animal that it's in that same breeding group. And so then you're not really changing the risk profile of that animal. It's still, and so like a lot of this, the reason I built my facility as a educational facility is that I have a commercial facility as part of it. And as part of that, I have, okay, well, here's how I minimize risk, which is not to say I get rid of it entirely, but to say that like, all right, I swap out gloves between racks. I sanitize between breeding groups, but within the same breeding group, I treat them as if they're basically the same animal, because they're all getting bred the same and they're all going to have the same microbiome, whether it's a virus or a bacteria or we really don't know, because the list is probably, endless.
That makes sense. So you mentioned losing like $250,000 of bloods. So talk about that experience and kind of what went down with that.
Yeah, well, so this was back in, about 2017, 2018. This was right when those viruses were really first starting to be described and we were first starting to understand them. And so a lot of python species are known for having seasonal resp. And when you hear about seasonal resp, a lot of times it turns out it's just these low-level serpentoviruses that pop up in stressed animals during breeding season and then go back to normal when the breeding season is off. And so green tree pythons are notorious for having this as a feature of breeding them. Lo and behold, they end up as a very high reservoir for many serpentovirus species. Blood pythons also have this reputation where they can frequently develop seasonal respiratory infection. And so, that was something that is a hallmark of many blood python colonies. And for that reason, there's kind of already a base level of virus in the colony. Then I brought in a new collection and did not quarantine it for as long as I should have. And one of those animals developed clinical disease, and then that clinical disease spread very, very quickly. After that kind of flash, I ended up moving part of the colony to section it out, and that applied kind of an even stress pressure across the whole colony. And I noticed that those low-level seasonal resp started developing real resp. and then disease just kind of popped up in a lot of stuff. And so like it's a combination of bad biosecurity and stress and biology of the animals, where basically like at that point the best knowledge we had was that virus positive is death just at some point. We really didn't have a complete understanding of how these might affect wild populations and why sometimes it's related to disease and sometimes it's not. And we still really don't necessarily have a great understanding of that. But basically, at the time, I just euthanized anything that was positive, anything that was with anything that was positive ever. And so I took the hard line, and I basically ended up culling out the entire colony. Or the only thing left was neonates that were not in the room at the time. And so what I have now is basically that. But even still, that colony, that is now the virus neutral colony, because those animals, which were never exposed to anything, kept closed for 5 years, still managed to have a brand new genus of virus. And so like, you know, we really don't know. We don't think there's vertical transmission, but we don't know that there's not vertical transmission. And so like, you know, everyone's just doing their best with the best information they can. At the time, a positive test or even anything that looked like it could have been exposed to a positive test, I took as grounds for euthanizing. Now the way I manage it is different, but I'm also operating with a different understanding of it. And I'm also not seeing disease relating to these viruses. And that is the big thing too, is that, you know, I do test and I do go through a very long quarantine. And even though I have animals in the colony that are managed very specifically that I know are low-level positives, I don't see disease really ever. And so all these different buildings have different risk profiles, and these different risk profiles are kind of my own little mini experiments to see, okay, what happens if I manage a collection in this way? What happens if I take it to as extreme as I can? And so like, I don't have all the answers, but I am certainly setting up different scenarios for my own stuff to see what these answers are, because I totally understand that, I don't know. It's a very complicated question, and we don't really have the answers. We're just doing our best to experiment our way through them. But yeah, so for me, a stressor is shipping is a great stressing thing to bring low-level virus to the surface, and then breeding is also a great stressing event that seems to bring low-level virus to the surface. And we also see this in wild populations too, where we found way more virus in the winter than in the summer in the Burmese pythons.
So technically, I mean, I'm not, but I won't say technically, but kind of like, I mean, basically what you're saying is your husbandry and the way you keep these animals is very important when it comes to whether or not you're going to end up with a sick population of animals. Correct.
Yep. Yeah.
Now,
yeah.
And oh, go ahead, because there's a follow-up part to that, but go ahead.
And you know, it probably seems like I'm like a lot of this is very complicated, and a lot of it is, but for me it also kind of gets simple too, which is that like the longer you give a quarantine and the longer you don't see any problems at all in that quarantine, the less likely there is that animal is going to introduce a problem into your population. And so incorporating testing into that is great, because testing is a fantastic tool, and it's great for answering very specific questions if you have very specific, you know, things you want to know. But at the end of the day, it's like, if you invest the time into making sure an animal is going to be healthy in the conditions you provide it, it's probably not going to cause problems in the conditions you provide it, you know?
Right. And now with those conditions, because I know a lot of the people in the comments right now are maybe like ball python breeders and things like that. And the majority of us, we keep our animals on cocoa, paper, things like that, in the tubs, room at a certain temperature. Do you find in your experiments with it, that something that it's easier for something to like breed a virus on, say, like a cocoa that some people might leave in their enclosure for months at a time versus a paper that's getting
It's really hard to say. It's really hard to say because you're also it's a very difficult, multifaceted question that goes into answers, areas we just don't fully understand. Because like when you are keeping an animal on paper substrate, it is more sanitary. But in being more sanitary, are you compromising the immune system of that animal and making it less likely to resist a pathogen? So like I don't know. Like these are just like this is how much we don't understand in terms of like the answers I think people think we know. Like there's really just a lot that we just don't understand, and people think that there's a rule book as far as the best way to do this, but like the berms in the South Florida aren't reading the rule book and they have virus. And so like, it's hard. It's a complicated thing to talk about, but also at the same point, it's like healthy animals oftentimes don't cause problems. And if you're using good biosecurity, and if you can afford testing and use testing in a way that is appropriate, then you're really investing well into a long-term colony. Ball pythons are actually a little bit easier in that the range of viruses we see in them are more limited than what we see in other species. Take that with a big grain of salt, because that is like our current understanding, and that current understanding is liable to change. But like a lot of times I caution people away from the cheaper tests, because the cheaper tests are more restrictive in the types of viruses they can detect. And their diversity is so high that people do a test that's really testing for this, and they think it's testing for all of this. And so they are operating thinking that they have this much safety when really they have this much safety. With ball pythons, this much safety might actually be all you need, because the range of viruses we see might fit kind of within that. And so like the cheaper tests, when done correctly, might be enough to like be a good tool for detecting virus in ball pythons.
So what reptiles are kind of on that wider spectrum that you would need like the more expensive tests? Like which ones are?
It's really hard to say. Yeah, because I mean, green tree pythons seem to be an obvious one, because about half the viruses we see are green tree pythons. But like, we also don't really know what we don't know. And so like, I'm only describing what I've seen so far. And so like, it might just be that like our test doesn't like ball python virus 2. And so there's a whole genus of ball python virus floating out there that I don't even know exists, that I'm not even talking about when I'm not, like, again, we just, there's known knowns, there's known unknowns, and there's unknown unknowns. And, a lot of the map is still very much uncharted.
How many times should testing be done for an animal that's in quarantine?
So again, it really depends on your risk threshold and your risk appetite. Like if you are, so the gold standard, what I did, if I said I want a building that is as close to NIDOVIRUS free as I can guarantee to be, I did 3 tests over the course of a year. So I did 1 test on receipt, 1 test halfway through quarantine, and 1 test to exit quarantine. And so that is the testing regimen that I used, but that was very, 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 very expensive. And so, it's realistic. It's like, 1 test is better than no test. 3 tests is better than 1 test. A really good test might be a great choice for something where you're worried about a diverse panel of viruses. A really cheap test might be, more restricted in what it can look for, but if it's looking for the right thing, then maybe that's fine. And so, it's almost like, I mean, again, you can do the best what you can with the tools available to you. But
and your quarantine right now and your quarantine practices that you're describing, so you're going a whole year is your quarantine?
Correct. Yeah. So I open the quarantine
I'm
for
slacking.
a certain amount of time, get in animals, close it, and it is closed for the year.
Oh yeah, I'm slacking, man. If you go on a year, I'm far behind a year.
Yeah. I mean, again, I've never seen a disease come out of an animal that has made it through that.
Yeah, that would make sense though. That would make sense for sure. And so do you what's the question I was about to ask? I forgot. I'm sorry. So as far as like when an animal tests positive for nidovirus or any of the viruses, excuse me if I'm saying like the wrong name or whatever, but
Yeah, like I said,
when an animal,
it's fine either way. Yeah.
when an animal tests positive, what's the next move for the average breeder? Like, what do you suggest?
So that is something I'm still working on having a better answer for. And so we did some experiments with antivirals. We've done some experiments with heat treatments. Some of that is looking promising. Some of that scares me. And so that is something we're still very much trying to investigate to make sure we're putting out good information for people, because there's also like viruses are moving things. And so, I really want to make sure that these treatments or whatever I try and do is truly getting rid of virus and not just pushing it outside of our ability to detect it,
Gotcha.
Like,
So
it could give you that same sort of false sense of security. That being
And when
said, treating secondary symptoms, a lot of times there's a secondary bacterial infection. And so treating with an antibiotic can be very beneficial. Increasing the heat can be beneficial, but also getting it out of the primary collection where it really can't do damage is, from the bird's eye view, just as important. Yeah. So, yeah, if we're adding stressors during quarantine, not necessarily. I've thought about that as well, as if it's worth stressing an animal to bring like low level virus to the surface. But I think shipping is a lot of times enough of a stressor. And then just being someplace new, eating someplace new, doing something new, like I think that's enough.
What were you about to say, Matt?
No, but he pretty much explained it. He said as far as raising up the heat, because when he said heat treatments, I was thinking like, are we talking like, because everything you're saying, I'm kind of just in my brain, I'm relating this back to people, because it all sounds like people to me. When we get stressed out, worried, we get sick. Our immune system drops lower. We get sick. You know what I'm saying? We get seasonal colds. We get seasonal flus. All it sounds like people. And then I know when I get sick, first thing I do is I go into the sauna and I'm trying to, you know what I'm saying, sweat it out. So when you say heat treatment, that's what I was thinking. Like, so are you taking your snake, maybe throwing it in the incubator, or your reptile, whatever it is, at a certain temperature to try to, you know what I'm saying, just increase that body heat to better fight off the virus? Is that the concept of it?
Potentially, but there's also a lot of there's a lot of things that make me nervous about that kind of stuff. So yeah, that is the kind of stuff that we're trying to work through, but there's a lot of reasons to not try and go crazy with it, because it just, like I said, these are very complicated things. And so we're trying to figure out what the actual benefit is to a lot of this stuff. But at the end of the day, like, a lot of the mechanics are, when we get sick, we get a fever, and a lot of times just increasing the metabolism is enough, along with antibiotics, to help these animals get over some of this illness. And once they kind of pass that hump, a lot of times they, again, I don't know what to do with those animals, but it isn't virus equals RI. It's a little bit more continuous than that. Yeah. So Billy's question about like, if they're doing a quarantine and they get more animals, the quarantine resets. And yeah, that is like, that is why I have a window in which the quarantine is open and stuff is getting tested as it's coming in, and it almost goes through a pre-quarantine. So like, I have something come in, it spends the first week in a bathroom waiting for the first round of negative tests. If it passes that first round of negative tests, then it makes it into the true quarantine. A certain number of animals will enter the colony in that window of like maybe 2 months or so. All of them, first have to pass a negative test. then go into that quarantine. And then after about, after I get whatever the species I want for that year, quarantine shuts, and it's multiple, basically until the calendar year flips around again, that I will consider getting anything new to the colony. And so, it is, but yeah, like people, when you get new animals, the quarantine resets. And if you are exposing animals in your quarantine, you are exposing animals in your quarantine. And so, like, for me, it's all about batching risk out. And so they pass the first sweep. Now they can all enter the quarantine together. And if I lose everything in quarantine, that would be horrific, but it wouldn't be losing everything in the colony. And I have a whole year to find out that I'm not going to lose everything in the colony, So it's like people want perfect, and perfect's just never going to happen. And so it's like just working with the best understanding and tools you got. And so it's like trying to come up with the best way to do stuff, and then trying to come up with like what's realistic within that.
Yeah, that's what I do for my quarantine. Like in an instance where I'll get like multiple animals in from like different places. If I always go through quarantine, I got limited space, and so like I got one place for quarantine. But let's say I have something in quarantine for like maybe half the quarantine time, and I get something new in, I got to put it in that same quarantine room, but quarantine starts at like day 1 again, and it's got to go through like that whole cycle before I feel comfortable bringing it into the collection. That's how I've been doing it. I don't currently test for like any of the viruses or stuff. I have a couple times, but it's not right now something that I'm doing like each and every animal that I bring in, but I do quarantine. But it's good to learn this stuff and really kind of learn about some of the risk mitigation stuff that you can do to kind of help yourself. Because if I'm hearing you correctly, what it sounds like is most collections have some sort of virus in them. It's just the level of the viral load that really kind of makes a difference between a sick animal and an animal that's really kind of asymptomatic. And it's those animals that have the high viral load, the viruses start multiplying, growing exponentially, that are more likely to pass that stuff on to your collection, get your animals sick, where you have like your whole collection drop out. Is that really kind of like
that is my best understanding of it, looking at everything that I've experienced, everything that I've done in the lab, everything I've seen in the wild snakes, everything I've seen in the 15 years I've been in the colony, in the private trade at every level, from 10,000 plus snake colonies to 100 snake colonies. Like, that is my understanding of what I think is the most likely scenario for how this is going down. And yeah, it fits within what we see, which is that we see a lot of virus, and we also see a lot of entirely healthy colonies out there that
So keep them healthy, keep them strong, they'll be all right.
Yeah, exactly. Yeah, just be on the lookout. If people who spend time with their animals and spend time working them and understand and pick up when a problem is going on and isolate that problem, just don't get problems.
Right.
Yeah, that makes sense.
Yeah, I just think, like I said in the beginning, or I don't know if I said it. Yeah, in the beginning, it's just been a lot of people just revving the whole virus thing back up that I've been seeing, and it's it's understandable to an extent, because I do. If you let it go, then and you're not taking care of your animals, you're not on top of it, then it'll destroy your collection. But everything you said, like I said, and my brain, it just keeps playing back to us as people, as humans, like taking the kids to the doctor. I give them vitamins. I make sure they're good. If they start to get wheezy, I make sure that they put the humidifier in their room, whatever. You know what I'm saying? Take those precautions if something little comes up. That way it doesn't blow up into something big. So this has been good, man. I appreciate all this information for sure.
Yeah, me too. And it's good to hear it from like someone who's spent a significant amount of time like studying this. And you said behind backstage that you grow more NIDOVIRUS intentionally than most people, like just in doing the testing and stuff like that. So it's definitely good to hear somebody who's very knowledgeable speak on it. Because I know for me, man, once you hear it, like Matt was saying, you kind of go through waves of hearing about it a lot, then it goes down, then you hear about it a lot, and so it's good to have a little bit more clarity because it's scary.
I should actually probably take the time to actually go over just some basic biology facts that we were able to discover
Okay.
working with these viruses in the lab. And so what I mean by that about I've intentionally grown more nidovirus than anyone else in the world is that like, so we have these reptile cell lines. And so one of the things I did for my research is studying the genetics of the virus, and that's like genetic sequencing and stuff like that. But that can only tell us so much. And so one of the things we want to do is grow it in the lab and figure out, you know, all sorts of things, what can kill it, how long it can last in the environment, stuff like that. And so one of the challenges is that like you need snake cells to grow snake viruses, and there's really not a whole lot of those out there. And so the lab that I was in worked with more reptile cell lines than like anywhere out there right now. And so, you know, the joke was that I had like an incubator zoo at the lab that was almost better than my collection at home. But from that, we were able to grow 5 different serpentoviruses or nidoviruses. And from those 5, well, so we developed a certain type of assay that is based on diamond python heart cells.
Okay.
And the virus kills those cells. And so based on the concentration of virus, it will either kill a group of cells or not. And so based on if the cells die, I can determine the concentration of virus, of live virus in the sample. And so we have 5 viruses that we were able to grow in culture. 3 of them killed the cells in a way that would work for this assay. And so that just shows you the diversity of these viruses, is that like we grew 5 of them, but only 3 of them worked in our assay. So everything I'm saying about these viruses are true for 3 out of 5 because we couldn't test the other 2. And most of the viruses we can't grow at all. So grain of salt in terms of what I'm saying. But for what we were able to grow, it looks like at room temperature, it'd only be able to last in the environment from a week to 2 weeks. If it's warmer, like 85, 90 degrees, it would be like closer to a week or so. But we start seeing dry virus drop only after a few days. So like if you have a sick snake and you have a cage and you disinfect it and you're worried, is there still virus in there? If you're waiting 2 weeks, the answer is like, no, because it'll just degrade in the environment on its own. Additionally, we tested a bunch of sanitizers against it and found that pretty much any sanitizer you use will get rid of nidovirus within about a minute. If you really want to be sure, you can do 3 minutes. And so bleach worked, ethanol worked, F10 worked, ammonia or peroxide worked. I forget what else we used, but basically these are not tough viruses. They don't last super duper long in the environment. They're very easy to kill with sanitizers. And so like, these are manageable things, like, you know, a good level of biosecurity, just washing your hands, like that kind of stuff, like preventing cross-contaminated, like just basic biosecurity. These things are very susceptible to basic measures in terms of preventing virus and disinfecting stuff. Versus like, I do work with other reptile pathogens in the lab that truly scare me, that don't get killed by sanitizers in the same way, that last way longer than the environment. Like there's actually really scary pathogens out there, but serpentovirus isn't necessarily in that same category. Like there's scarier things out there, but I also say that wanting to make sure it doesn't sound like I'm minimizing it. And I also want to make sure it doesn't sound like I'm like saying don't test your animals or testing isn't important or minimizing the value of testing. Like it's a very nuanced conversation, which is that testing is a tool and that a lot of this is complicated. And at least serpentovirus is one that is manageable.
Gotcha. So I had one question on deck for you, and that was with regard to like shows and letting people hold your animals at shows. But now also after you said that, I got another question too, as far as like the pathogens out there that you said that are scarier. Can you speak to some of those too after you chat about like best practice at shows and stuff like that?
Yeah, so really it just depends on the type of pathogen. And so like serpentivirus is an enveloped virus. And so that means that there's a little layer of fat that surrounds the virus. And if you break that layer, the virus is basically inert. There's other viruses out there that don't have that same fatty membrane, that are just basically tight balls of protein. And those are much harder to kill. And so like that kind of stuff is scary. Crypto is not a virus, but that's another thing that is like very hard to get rid of, that like a lot of things don't kill crypto. And so like there's very scary things out there for sure.
And do those with those because do you, as far as yours go, you have a lot of different animals and stuff. Do you keep different species in separate rooms just for that, because a lot of them carry different things or whatever? Like I'm interested right now. I'm putting, like, I have other animals. I got a tegu, I got a caiman lizard, I got a boa, I got a retic, and a burn, right? But I'm interested. I'm thinking about, once I knock this wall down, putting all those animals, even though they're all healthy, into the same room as the ball pythons. Is that something that you would be like, no, those guys carry something that'll wipe out your whole collection type deal?
No, not particularly. I mean, again,
Okay.
it's like, it's just like there's like there's general statements I can make, and I can go and find a specific answer or a specific example that would probably show you the opposite, which is like, you know, like generally the more divergent something is, the harder it is for it to jump hosts. So like the serpentiviruses, we really don't see like them jump between genre of reptile typically. Like this is the python viruses, these are the, you know,
Okay.
colubrid viruses, these are the something else. And so like oftentimes the more divergent the host, the harder a pathogen will have to jump between it, except for when that doesn't happen. And then a lot of times you end up with something that is in something it shouldn't, and ends up just going completely berserk. And then you end up with like a spillover event, and then you can end up with problems. But again, it's not like it's like predicting lightning. Like I can give you general rules, which is that like long quarantines, and you know, the longer quarantine the better. Like for me, a year is great, but like 6 months is like, I don't know, people do like as low as a month, and that's just it's all about risk profile. And so like it's hard to make any rules for anyone. It's more about just what you're willing to accept for a risk profile, and just understand that like a year-long quarantine is twice as good as a 6-month quarantine. If you can live with that, then you can live with that. And that's, I don't know. But shoot, I lost my so generally speaking, the wider diverse, you know, the more divergent the hosts are, the less likely there is to be problems, unless there isn't. But for the general rule, you're pretty safe. I keep a lot of my species together, but you know, I also like go through a year-long quarantine and stuff like that. So it's you know, it's different, and it's also just a different setup. Like I understand that what I'm doing is probably never going to be fully perfect, and if I end up with a problem, I have, you know, risk spread out within that. And so like I have multiple buildings, and so if I have a problem, it's going to be limited to just one building. Within that one building, I swap out gloves between racks. And so if I'm going to have a problem, it's probably just going to be limited to one rack. Within that rack, I have stuff broken into breed groups, and I sanitize my hands between breed groups. And so if there's a problem in a rack, it's likely to actually just be limited to just one breed group. And so like the amount of barriers a pathogen has to break through to, like it has to pass through multiple layers of biosecurity without me noticing it before it would actually blow up into a problem. And so, like, again, it just that's a complicated answer. And it's like, if you keep multiple reptiles in the same room, that is more risky than having multiple reptiles in different rooms. But that's not going to keep me from having multiple reptiles in the same room. Like, that's a risk I'm willing to accept.
Right.
Because, you know, I invest in making sure they're coming into that room healthy, because I'm banking that hopefully if there's a problem, it'll show up in the quarantine.
That makes sense. That makes perfect sense.
But, like,
So,
you know, yeah, again, someone else might have a different risk profile, which is to say that, like, I really need this animal into my colony as soon as possible. And so I'm going to knowingly put it through a poor quarantine, but test the snot out of it and hope that that is, again, good enough. And so, like, you know, if you are sacrificing quarantine with good testing, but even still, like, that's never something I would recommend doing. I'm just like, you know, it's not there's no rule book on it, and people expect there to be a rule book on it. And so it's, yeah.
that makes sense.
No, what you're saying is good. Yeah, that's good info. You know, kind of almost make it dummy proof. You know, so.
Yeah, well, I mean, like, at the end of the day, I live in the real world, and, like, people got to sell snakes, and, like, you know, testing is expensive and quarantine isn't. So that's been my solution. But at the same time, like, there have been other parts of the colony at other times that I have invested in testing heavily, because, again, I have a building with half the world's python species in it that I know are as nidovirus free as could possibly be, because I spent just as much money testing those animals as the animals themselves. And so, like, but again, that does not work on a commercial basis. Like, so that's why I have a commercial building where I did a different risk profile. And, you know, yeah,
that makes perfect sense. So, are there any other, like, tips or recommendations that you could give us and give the listeners in this space?
Again, for me, the base part of it is that it is impossible to culture any organism without also culturing its microbiome. And so I've done a lot of cell culture work, which is we're taking a certain tissue collected under the most sterile set of circumstances possible, put in the most sterile incubator possible, kept in the most sterile way possible. And there will still be some sort of bacteria or something that hitchhiked along, because the microbiome is part of an organism. And so it's important to understand that, like, it doesn't need to be zero virus to be a healthy animal, and that this is just what we know about today. But, like, if your expectation is zero problems, you should not be in the animal business. Animals have problems. And so, yeah.
Yeah, that makes sense, man. It's inherently going to be some risk. It's inherently going to be some problems, man.
Yeah, like you don't want a sterile animal. A sterile animal is a bubble boy. Like, that's how you get no immune system and you die. So, like, you know, this is a nuanced conversation, and, like, you don't want an animal with no microbiome, and sometimes viruses are a part of the microbiome. And so a lot of times it's a conversation about managing risk and what to do when problems do occur, and what to do to prevent problems from having the least likely chance of occurring in the first place. But there's never going to be, like, do this, 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 and this, and then you're golden, you know?
That makes sense. Matt, you got any more questions?
No, man, I don't. All my questions were asked by the viewers on here, and or he already said it. So, man, I've soaked up a lot of information. This is good.
Yeah, you covered every one of my questions that didn't get asked. That was dope, man. I think a lot of people gonna look back at this and see this because, again, it's something that needs to be heard. In terms of like just clearing up a lot of the misunderstanding out there and adding a little bit of clarity to the situation. Obviously, we're not at a point, and probably never will be at a point to where we got all the answers, but at least kind of having some guidelines and some in a common sense, practical way to think about it, particularly as breeders.
In some ways, I almost feel like I've removed the clarity or muddied up the waters in a lot of ways, in the sense of like, it seems like people talk with so much certainty now. Like people talk with the clarity that like you did something wrong to sell a sick snake or a virus positive snake, and it's like, no, you can do everything right and still end up with a virus positive snake. And so it the certainty that people have when they're just throwing each other under the bus and going at each other's throats, you know, I've done everything I can to study these questions as deeply as I can, as extensively as there is to know. And I'm telling you that I don't know the answers to these questions. And so if anyone out there is saying that they do, like, okay, fine, you know.
Right.
You saw that too about him sitting, even trying to wipe that black spot.
I even tried. I'm so sorry. Yeah, I tried to wipe it off my camera. I don't know what the black spot is.
I thought it was on my screen.
I thought it was my iPad. I thought I was the only one.
I thought
No,
I
I thought it was me first. Yeah, I apologize. I don't know.
Oh no, you're good. You're good.
Yeah, I can tell you it's not
I
on my
did
camera
before.
lens because I wiped off my camera lens. So.
Yeah, it's all good. I done been there with stuff like that before, but I was like low key trying to get it without nobody seeing.
Yeah. Oh man.
Man, this has been a dope episode, man. We really appreciate you letting us know, man, giving us the 411.
Yeah, absolutely. And I know you're in the area. At some point, you know, you really need to come and check out the facility, because it really is just like, I just posted up a facility tour where I had some educational groups out, and it really covers the expanse of the facility well. But it's like I said, any idea I had, I just took it to 10. And so a lot of this is saying, based on what I've seen, how am I operating in the real world? And so like it really is almost as a how to deal with NIDOVIRUS facility in some ways. So
how far are you from Daytona?
About 2 hours.
Yeah, you're probably about 15 minutes away from me, if that.
Yep. Yeah, I'd imagine. Yep.
Yeah. And it's crazy.
I've got an extra day out there, so might have to try to bribe the wife into doing something. We'll see.
You stop to the facility too.
Right, you know what I'm saying? Yeah, we might have to.
Yeah, you can do a 2 for 1.
Shoot.
Yeah, man, it's been good, man. Really appreciate it, man. Definitely appreciate everybody coming through, man, showing love in the chat, asking some great questions, man. Definitely share this out, man. Tell people to come get some good information and some information that can clear stuff up for them a little bit and kind of get them like some guide principles and some practical advice, particularly for what we do.
Yeah, at some point I should come on and talk about some of my other stuff too. I've been doing reptiles for a very long time. So I've done some trips over to West Africa, and so like a lot of the ball python genetic tests you're seeing now, like the genomes that those are based off of, those are ball pythons that I like brought back from West Africa. And so like I have a very eclectic collection that covers just stuff that you don't see elsewhere, and so it very is a unique. Yeah,
that'll be dope, man. Like I don't know like what your protocol is or whatnot, but if you're open to it, maybe we can do an on-site show.
Yeah, sure.
Cool. All right, man. Well, again, man, appreciate you coming out. Appreciate everybody in the chat. I think that's gonna do it for us tonight. Y'all make sure after tonight's show, you go check out Sophisticated Ignorance. Getting ready to get started in just a few minutes over on the homie Malacio from Wiz Constrictors over on his channel, man. They always get sophisticated, they always get ignorant, a lot of good time, good laughs, and all that good stuff, man. Appreciate y'all coming out. That's gonna do it for us tonight, though, man. For Double Clutch, I'm Mike. That's the homie Matt. This is Stephen Tillis. Go follow him on social media. The links are in the description of the video, so definitely go check him out on social media. Hey, but we're gonna be out for tonight. Thank y'all.
Hey, thank y'all, man.
Yes, sir. Be blessed.
Thank y'all.
Peace.
With Mike and Mac, we lead the way. In the reptile world, we're here to stay. Double clutch where passion meets. From the start to the top, you can't be beat. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. With Mike and Mac, we lead the way. In the reptile world, we're here to stay. Double clutch where passion meets. From the start to the top, we can't be beat. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Always on track. With Mike and Mac, we lead the way. In the reptile world, we're here to stay. Double clutch, where passion meets. From the start to the top, we can't be beat. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Always on track. Always on track. With Mike and Mac, we lead the way. In the reptile world, we're here to stay. Double clutch where passion meets. From the start to the top, we can't be beat. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. With Mike and Mac, we lead the way. In the reptile world, we're here to stay. Double clutch, where passion meets. From the start to the top, we can't be beat. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Double clutch, with Mike and Mac. Double clutch, it's where it's at. Double clutch, we lead the pack. Double clutch, always on track. Always on track.